In the last video, we finished up explaining briefly the difference between the humoral and cellular branches of the adaptive immune system. So I want to go into a little more detail of that, and then we're going to, in this video, focus on humoral. Next video will be cellular. So this is a more detailed step-by-step process by which the humoral and cellular adaptive immune systems are activated and fight pathogens. We will go through all the steps within. So we're going to start with these bullet points, and then we'll go through in more detail another time, a second time. So the humoral immune system involves the process of antibody production. When B cell receptors bind an antigen, the B cell will phagocytize the pathogen. Upon phagocytizing the pathogen, it will become activated. And what I mean by activated, I mean it's going to proliferate clones of itself with the exact same B cell receptor that was able to bind to that antigen on that pathogen. So you have a B cell encountering a pathogen. The B cell receptor on the B cell binds to the antigen on the pathogen. The B cell is then stimulated to phagocytize, ingest and digest the pathogen. The B cell becomes activated, and it starts rapidly producing clones of itself, true clones. So before the B cell was ever activated, we call it a naive B cell. So when naive B cells replicate, they don't make true clones. Each daughter cell is going to have a unique, randomly generated B cell receptor. But once a B cell becomes activated, it is no longer naive. When it replicates, it makes precise clonal replicates of itself with the exact same B cell receptor that binds to that pathogen. Some of the daughter cells are going to be long-lived memory cells. They're going to have that B cell receptor that attacks that pathogen, and they're going to sit and live in your body for years and years, maybe even your whole life. So what you've done now is you've generated an army, a long-lived army of B cells devoted to fighting that exact pathogen. That's what gives you immunity to that pathogen. Other daughter cells are going to become antibody generating factories. What the antibody generating factories do is, remember I said the B cell receptor is the same as an antibody, except the B cell receptor is in the B cell, the antibody is excreted into the body fluids. So antibody factories just spit out massive, massive numbers of that B cell receptor, now it's called an antibody, into the body fluids. And those antibodies, when they're circulating, they are going to, even though they're not attached to a cell, they're going to bind to and attack that pathogen that they're designed to bind to. So the antibody generating factory cells, they are not long-lived. They wear out real fast. They're spitting out thousands of antibodies every second, every minute, and they don't live very long. So they wear themselves out real quick. The memory cells are going to live for years and years. The B cells start out as stem cells in your bone marrow, and they will mature in your bone marrow, and then they'll be sent to the lymph. And so they spend most of their time in your lymphatic system. And these B cells, they're best at, so this whole process, the humoral immune system, B cells, B cell receptors, antibodies, what they do is they mostly fight toxins that are circulating in your body fluids. They fight viruses that are still circulating, so the virions, not the ones that are inside your cells, but the ones that are in between cells, so the virions that are in passage from one cell to the next. Most bacteria, protozoa that are floating around in your body fluids. So that whole process is very similar to how T cells work in the cellular immune system. So remember, the cellular immune system specializes in attacking pathogens that are trying to evade immune responses by entering and hiding out inside host cells. T cells do not excrete antibodies, but instead use cytokines to help destroy pathogens. They are coated in T cell receptors, which are very like B cell receptors. They are very specific to a specific antigen, but T cells bind to antigens that are displayed on MHC molecules. Different T cells will bind to different kinds of MHC molecules. Remember, MHC stands for Major Histocompatibility Complex. So I told you that all cells in your body have MHC class 1s. Those are usually used as a self-tag to indicate I belong here. If any one of those cells of your body becomes infected with a pathogen, it's going to place an antigen from that internal infection in its MHC class 1. So normally it's a name tag saying I belong here, but in case that cell, that host cell gets infected, that human cell gets infected, it will place an antigen from the internal infection in the MHC class 1. A T cell comes along, it checks all the MHC class 1s to see if they're normal. If it finds an antigen, it is going to destroy the infected host cell. When it destroys the infected host cell, it's going to expose the pathogen that was hiding inside of it. There are other T cells that bind to MHC class 2s. MHC class 2s, remember, are only found on antigen-presenting cells. Antigen-presenting cells are cells that phagocytize a pathogen and then present the heads of its enemies to the T cells. So T cells go around, there are some T cells that go around checking MHC class 2s on all the antigen-presenting cells, which are part of the immune system. If they find an antigen in the MHC class 2, it knows that that antigen-presenting cell phagocytized an invader. The T cell becomes activated and it releases cytokines to activate all the other T cells. So again, we will go over that in the next video, the cellular immune system. But the main thing that you need to know is that T cells, they have those T cell receptors that are highly specialized for specific antigens from specific species of pathogens, but they find those antigens in MHC molecules presented by your own host cells. And they focus on fighting intracellular pathogens. So they're attacking antigens within host cells that's mostly going to be viruses. There are a few bacteria that can enter host cells as well. T cells can also identify abnormal host cells like cancer cells. So they also play that role in destroying cancer cells. T cells start as stem cells in the bone marrow, but then they travel to the thymus and they mature in the thymus before they're released into the lymph. And if you don't know where the thymus is, it's here. So it's sort of like positioned between your two lungs. All right, so for the rest of this video, we're focusing on breaking down that humoral immune system pathway even more. So immunoglobulins. So the first step is an immunoglobulin binds to an antigen, and that forms an immunoglobulin antigen complex. The immunoglobulin, Ig, is either a B cell receptor or an antibody. Those are the two kinds of immunoglobulins. Remember, an immunoglobulin embedded in the B cell surface is a B cell receptor. An immunoglobulin that's released by the B cell into the body fluids is called an antibody. So you have an immunoglobulin that binds to an antigen on the surface of a pathogen. Now it's an immunoglobulin antigen complex. So that's step 1. Step 2, the immunoglobulin antigen complex binds to the B cell if it's not already bound. So if this was an antibody floating around in the bloodstream, the variable region, the epitope, the paratope binds to the antigen, right? And then the stem has to go back and bind to the B cell again. So the stem is the constant region. So the immunoglobulin antigen complex binds to the B cell if it's not already bound there. That's step 2. Step 3, the B cell is now going to phagocytize the antigen and the microbe that it's attached to. So once you have this immunoglobulin antigen complex on the B cell surface, it stimulates the B cell to ingest the antigen and the microbe that goes with it, and it digests it. So that's phagocytosis. Next, so B cells are a kind of antigen-presenting cell. So they're going to display the fragments or the antigens from the pathogen they just digested on their MHC class 2s on their surface. So this part I think is really confusing to students because you had B cell receptors on the surface of the B cell. You also have MHC class 2s, which is another kind of receptor on the surface of the B cells. Remember that all cells have many, many, many receptors on their surface for all different kinds of signals. So we're talking about 2 of them now. The B cell receptor binds to the pathogen. The MHC class 2 presents the fragments, the digested pieces of that pathogen to other immune cells to show other immune cells what they did. So step 4 was display fragments or antigens on the MHC class 2s on the cell surface. Step 5, a helper T cell binds to the antigen on the MHC class 2 cell surface and is activated. So it produces cytokines. So just to recap, you had an immunoglobulin binding to an antigen and binding to the B cell. The B cell then is stimulated to ingest and digest the pathogen, but that's not enough. So now it's gotten rid of that pathogen, but that's just one, and there's probably more. So now the B cell needs to tell other immune cells about it. So it's going to present antigens from what it just digested on its MHC class 2s, and these T cells called helper T cells, TH, are going to bind, they're the ones that are going to acknowledge this pathogen. So helper T cells communicate with B cells. Helper T cells are going to bind to the MHC class 2s, and when they find a pathogen there, they're going to become activated and produce cytokines. All right, so here is a picture of this. So you have the microbe here, and this is an antigen presenting cell. We're going to say it's the kind of antigen presenting cell it is a B cell. So this B cell binds to the antigens on the surface of the microbe with the B cell receptors, causes it to phagocytize the microbe. The microbe is digested, so you can see inside here there's all these different fragments of the microbe. There's an antigen fragment there. The antigen fragment is then placed into the MHC class 2 molecule, and here's a helper T cell. The helper T cell can dock here on the MHC class 2 and check to see if there's an antigen in there. If there's no antigen there, the helper T cell thinks, oh, there's no invasion, there's nothing going on. But if there is an antigen here, what that tells the helper T cell is that this B cell has just digested an invader, and the helper T cell is then going to release cytokines. Those cytokines are going to activate an immune response. All right, so let's continue with our process here. So we're on step 5. Helper T cell binds to the MHC class 2 with the antigen and produces cytokines. Step 6, the cytokines are going to activate the B cells. So the helper T cell releases cytokines, that activates the B cell, it causes the B cell to proliferate. So I was telling you before that the B cells proliferate rapidly once the B cell receptor binds successfully to an antigen on a pathogen, the B cells proliferate rapidly. Well, these are the steps in between. So the helper T cell, it needs a co-stimulatory signal from the helper T cell. And that's sort of a checks and balances system, because we've talked about autoimmunity. That's when your immune system attacks your own body, your own tissues. But allergies are when your immune system attacks a foreign thing that's not pathogenic, like a harmless thing like egg white or pollen or peanuts. Those are not pathogens. They're not going to hurt you. But if you have an immune response, your own immune response could hurt you. So to protect against allergies, this whole checks and balances system, that's supposed to protect against allergies. So you're not constantly reacting to things that don't need to be reacted to. So it's not just the B cell by itself that has to be activated. It has to be the B cell plus the help of the helper T cell. So the helper T cell releases cytokines that activates the B cell. Then you get that rapid proliferation of the B cell. Some of the the daughter B cells are going to become those antibody factories, and then others of those daughter B cells are going to become the memory cells that protect you the second time you encounter the same pathogen. So here's a diagram of that. It doesn't show the whole process, but you can see there's a stem cell here, and it's showing the gene rearrangement. That's the mitotic genetic recombination. So these stem cells that are producing white blood cells, here's all the B cells that they produce. They're all different colors, and here's the B cell receptors on their surface. Their different colors are representing that they have different B cell receptors. So even though this is mitosis, which is supposed to be clonal cell division, you're getting genetic recombination and mutation in the genes for the B cell receptors. And so each of these B cells that are produced has a different receptor. Now, they're numbered also. So yellow is 1, pink 2, blue 3, green 4. In this diagram, B cell number 2, the pink one, it has a B cell receptor that could successfully bind to an antigen on this pathogen that's invading this human. So this B cell now is going to proliferate, and it's going to create exact copies of itself. All those pink ones are proliferating and making massive quantities of pink daughter cells of the B cell number 2. All of these immunoglobulins, the B cell receptors, the antibodies, they all have the exact same binding site for this pathogen. So some of them you can see here are these antibody factories. They've got a lot of rough endoplasmic reticulum. Remember, rough endoplasmic reticulum has ribosomes to read genetic material and produce proteins. Antibodies are a protein. So it's got a lot of rough endoplasmic reticulum, so it can produce a lot of antibodies. And then these other ones are these long-lived memory cells that stay in the body for years and years. So what's not shown here is the role of the helper T cell, which would mean that this is, so if a helper T cell was here, that would be normal. There are pathways that skip the helper T cell part. So there are certain antigens that do not require help from a helper T cell to initiate this process. So what we're seeing here is what we call a T-independent antigen. So it's called a T-independent antigen because it does not need to have activation from a helper T cell to stimulate the proliferation of this B cell. Okay. So some antigens can activate B cells directly in this way, but usually the B cell will need to be activated by a helper T cell before it can proliferate. All right, so just to recap one last time, because I know it's a lot, you have an immunoglobulin binding to an antigen. That's the immunoglobulin antigen complex that binds to the surface of the B cell if it's not already there. The B cell then phagocytizes the antigen in the microbe it's attached to. It digests it and presents the antigens from what it just digested on its class 2 MHCs on its surface. A helper T cell checks the MHC class 2, finds an antigen there, and so produces cytokines, which activates the B cell. B cell proliferates. Some of the daughters become antibody factories and others become memory cells for the second encounter. So we've talked about B cell receptors and what they do. They bind to an antigen and that stimulates phagocytosis. Phagocytosis destroys the pathogen attached to that antigen. But when immunoglobulins are released into the bloodstream as an antibody, what do they do there? What's the purpose of that? So there's 5 ways that a free-floating immunoglobulin, an antibody, can destroy pathogens. We have agglutination, opsonization, antibody-dependent cell-mediated cytotoxicity, which is a mouthful, neutralization, and activating the complement system. So we'll go through those one at a time. So here at the top of the slide, you see antibody-antigen complex. That means the antibody has bound to an antigen, which is usually on the surface of a pathogen. So what does it do then? Agglutination is when the antibody-antigen complex clumps together. So all of these antibody-antigen complexes clump up together. And one thing that agglutination does is because all of these antigens are now clumped together with their pathogens, it makes it easier for a lot of those pathogens to be phagocytized at the same time. So you can get rid of a lot of pathogens at once by clumping them up together. Agglutination is also what you see when someone gets the wrong blood transfusion from the wrong donor. So if your antibodies are accustomed to type B blood and you receive blood from type A, the recipient's antibodies are going to attack the donor's blood because it looks foreign. And what it causes is clumping up of the blood in the blood vessels, and that causes a whole lot of problems because of agglutination. Right, so opsonization. Opsonization it refers to facilitating phagocytosis. So there are some microbes that are really difficult to phagocytize. So for example, capsules, like we talked about with microbial mechanisms of pathogenicity. Capsules are protective because they're difficult to phagocytize. They're very large, which makes it logistically challenging for the phagocyte to reach its pseudopodia around. And they're also negatively charged. And because the phospholipid bilayer on the white blood cells is also negatively charged, it repels the white blood cells. When an antibody is attached to a microbe with a capsule, we say it opsonizes it, which means they kind of become like grips that make it easier for the white blood cell to phagocytize them. So opsonize means facilitate phagocytosis, but it's really because it's sort of like a grip to make it easier for the white blood cells to grip onto the pathogen. So microbe coated with antibodies to enable phagocytosis. All right, this one is the mouthful. Antibody-dependent cell-mediated cytotoxicity. A microbe coated with antibodies is destroyed outside the immune cells. So opsonization means it's facilitating phagocytosis, where the pathogen is destroyed inside an immune cell because it's digested. But sometimes a pathogen that's coated with antibodies will stimulate a different kind of white blood cell to secrete antimicrobials. So some of your white blood cells actually produce antibiotics. So you have your own endogenous antibiotics. So some of your white blood cells can't phagocytize pathogens, but they can secrete certain kinds of antibiotics, your own homemade antibiotics, and spill those antibiotics onto the pathogen. So those white blood cells that create those antibiotics, they are stimulated to do that when they find an antibody bound to the pathogen. So antibody-dependent cell-mediated cytotoxicity is when an antibody binds to an antigen on a microbe and it stimulates an immune cell to secrete all of these poisons that kill the microbe. Neutralization. Antibodies can prevent pathogen attachment to the host by neutralizing the adhesins. So we talked in microbial mechanisms of pathogenicity. Microbes have these adhesins, which are proteins that help them attach to host cells. When antibodies bind to adhesins, it neutralizes them so they can no longer bind. So that prevents invasion. Also, neutralization can refer to neutralizing toxins produced by the pathogen. So we talked about exotoxins in the microbial mechanisms of pathogenicity. So for example, Clostridium tetani produces a neurotoxin. When antibodies bind to that neurotoxin and neutralizes it, so it's no longer poisonous. Activating the complement system. We're not going to get to the complement system until the very end of both immunity chapters, but the complement system is a crosstalk system between the adaptive and the innate immune systems. Antibodies activate it. So it's just a branch of the immune system, and it's activated by an antibody-antigen complex. Okay, so going back to this step-by-step for the humoral immune system, we had step 1, immunoglobulins bind to the antigen, creating the immunoglobulin antigen complex. This binds to the B cell if it's not already bound. B cells ingest the antigen and the microbe it's attached to and digests it. I mentioned in one of the slides that so the next few steps are to activate the helper. There's co-activation between the B cell and the helper T cell. But I mentioned in a previous slide that some antigens can skip the whole helper T cell part. So let's look at that. For a T-independent antigen, step 4 would be T-independent antigens activate B cells directly. They do not need help from the helper T cells, so they just create proliferation right away. And then you have the B cells, some becoming antibody factories, others becoming memory cells for a second encounter. So that is the end of the humoral immune system. The next video will be the cellular adaptation.